Our speaker today is Manoj Gopalkrishnan. He will be speaking on life minus matter equals algorithms. Manoj will tell you more about it.
Hi. So the idea for the talk today is that I want to think of biological processes as performing some sort of computation, and I want to argue that the ways in which living systems are different from matter are due to the sophistication of the algorithms that they are performing. I hope you enjoy the talk. Let me just get started with my talk. So I'm sure all of you have seen these things before. They are crystals of sugar, mishri as you call them, or Culkin as we call it in Tamil. So here's a puzzle. If you look at these things, if you ever wondered about it, you see these straight edges, right? You see that there is sort of a straight edge here, there is a straight edge here. They seem to be at a nice angle, they seem to be around 90 degrees. So how did that happen? So it's not as if someone cut that sugar in that shape, right? You just put some sugar in water and you let the water evaporate, and in the process you find such crystals forming. So how does this happen? How does the crystal somehow know that it has to form these straight edges and it has to form these right angles? How does that happen? This is a three-dimensional problem. Let's try to look at a toy model in 2 dimensions. So let me say that I have a square. I have a square that can stick to itself north to south or east to west, and I have many such copies of this square tile. So I have this one square tile, another square tile can come in and maybe it will attach on the east of the first square tile. Another square tile can come in, maybe it will attach on the north, maybe another will come in here, another here, another here, another here, and go on. So right now there seems to be no reason to suppose that this will form anything nice, right? It just looks like some blob, it just forms something random. So what do we have to do to make this form something nice? So I'm going to impose one more condition. I'm going to say that if a tile comes in and sticks at only one position, then it's not very stable. It's going to fall off very fast. But if it gets the opportunity to stick where there is more than one attachment possible, then it won't fall off. It will stay there, let us say, forever. Now let us see what happens. So now if one more tile comes in, it's likely to go in here because it had 3 attachments here. And if one more tile comes in, it can go here because it had 2 attachments. And again, here there are 2 attachments, here there are 3, here there are 2, here there are 2, here there are 2, here there are 2. And again, here this row has started in some sense, so it will go on and it will zip up this row. And this row has started, it will zip up, and you start seeing these smooth facets, right? These smooth edges. Just because it likes growing where it is rough. Where it is rough, it grows faster. But this still does not explain one thing. If you look at these shapes, they look about the same dimension on length and width. And that's again not something that our model has told us so far, because it could be that this keeps growing, but it only keeps growing in one direction. It never grows in this direction. Maybe something comes and sits here, and as soon as this comes and sits here, this row fills up. Then something else comes and sits here, and then this row fills up, and so on. So why does that not happen? Because it's when a new molecule comes in, when a new square comes in, it's about equally likely to sit in any position that it finds. And as soon as this dimension, as soon as this becomes longer than this, then the new tile is likely to come and stick on the longer side. And so that balances out and you get things that are sort of symmetrical, about the same length and width. So the second principle we have learned is that it grows faster where it is thin. So in the end you balance out on your dimensions. So here's another cool thing crystals can do. Suppose I take the same sort of square crystal, and by some process it gets shattered, say along the diagonal as I have drawn in red here. Now when it gets shattered, I get these two pieces of crystal, and if more square tiles come, they are going to fill out these two pieces of crystal. So I actually get two copies of the crystal where before I had one. Now if each of these shatters, I will get 4 copies. If those shatter, I will get 8, and so on. So it is as if this crystal is self-replicating. It's breaking into pieces, and each piece grows into a complete crystal. Now when I talk about self-replication, one would like the copying process to be exact, and I sort of assumed that it breaks along the diagonal. Why should it break along the diagonal? Maybe it breaks along, some other line, and then I will not get very nice shapes. So perhaps I shouldn't be thinking about squares, but perhaps I should be thinking of something more like a ribbon. So let me try to draw something. So again I have squares coming in and forming something, and let us say that I have what is called a dislocation fault on a crystal. So what happens is the new tile comes halfway in between these two, and so it sort of sits out here. So what happens because of this is that now where will the next tile come and sit? It could sit anywhere here, it could sit anywhere here, but those are all single attachments. There is a point here where it has slightly more than a single attachment. It has like 1.5 attachments, and it's going to prefer coming there rather than sit on a single attachment because it will fall off faster there. So then chances are it will sit here, and then this row will fill out, and then the next one can possibly sit here, and the next row will fill out. So you see that having such faults can cause a crystal to grow faster. So it now grows out like a ribbon, and when I have a ribbon, it's easier to break a ribbon, so that however I break a ribbon, both the pieces are going to grow like ribbons. The break is also going to inherit this sort of fault, this sort of dislocation fault. And so in some sense crystals can encode some information, and that information can also be transmitted to the next generation of crystal. So this almost starts sounding a little bit like the theory of evolution, if you have seen those things. And indeed this person A.J. Kane Smith has a proposal that perhaps life began with crystals, and the particular crystals that he talks about are some crystals of clay that form on the banks of rivers, and these are of a very small dimension, not as big as sugar crystals. And that may or may not be the case, but it's at least sort of a plausible direction in which one can start thinking. In nature, so the sort of model that Kane Smith was proposing, there are many types of crystals and there are many types of molecules, and they are often specific as to which molecule likes to stick next to which other molecule. And so there is more information in these individual molecules. So let us see if we can incorporate that and get something more. So in this model, I have these 1, 2, 3, 4 squares, and you see that I have made these half circles on those squares. The half circles are meant to denote which other squares these squares like to stick to. The squares with dark shaded half circles, that edge likes to stick to other dark shaded half circles, and the light shaded half circles, they like to stick to other light shaded half circles. And I am assuming that I have some sort of L shaped already formed from some other tiles. In fact, I will need 3 other tiles to make this L shape. So this has already formed, and I am going to play the same game as before. So 2 attachments, it will stay. 1 attachment, it will fall off. So I will not even look at 1 attachment. Let me give these names. Let me call this tile 1, tile 2, tile 3, tile 4. So what tile should come in? So where will the tile come in and what tile will come in? I want someone from the audience to tell. 4 will come in where? Right. So let me just fill this out. So I fill this out here, and because 4 has empty circles here, I draw empty circles here. So I fill this out and I fill this out, and you see what I am doing, right? So then I do this, then I do this, then I do this, then this. So there is a pattern that will start to emerge, So you will get triangles within triangles within triangles and so on. So we started with a square grid. So of course these are not perfect triangles. If you go back to the previous picture, you see that these are triangles with rounded off vertices. But if you do this for long enough and you zoom out enough, then it starts looking like this. And the message is that these tiles, right, these simple games that we were playing with tiles, it can make these complex patterns. And so you can ask, what else can it do? What are the complex patterns it can make? What are the complex patterns it cannot make? In some sense, this is almost like a computer program, and you can make this precise. So the tiles with the glues that you put on them, they are like a computer program, and then you just follow those rules blindly. It's just a chart that you keep filling out, and in the end you get whatever you get. And it turns out that whatever you can do on a computer, you could as well have done it using tiles like this. So you can convert every computer program into a set of such tiles. And why is that interesting? One reason it's interesting is because all the laws of physics that we know of, all the laws of nature that we know of, we know that they can be simulated on a computer. But if computer programs can be done, in such simple models, that suggests that, this is quite a powerful model. And so we see such things happening in nature all the time. So maybe we should start thinking of that maybe nature is doing computation. Maybe that's a useful point of view to take about natural processes. What I am showing you is some recent work. These are microscope images of actual objects made out of DNA molecules. And each of these objects is very small. So each of them is about 100 nanometers on the length, which is about half the wavelength of visible light. So the smallest thing you have ever seen using an optical microscope, it's about half the size. So these are really, really tiny things. And obviously, since they are so small, you cannot see them with light. Instead, these have been imaged with a needle that, they have been put on a surface, and once they sit on the surface, a needle bounces over them, and depending on how much the needle bounces, you get out height information. And that height information has been encoded using false colors. So really all you have is height information, black and white, and we just put colors to enhance the contrast in that height information. So DNA is a string of molecules. So such strings are called polymers. And in the case of DNA, the individual units in this string come in 4 varieties: A, T, G and C. It occurs in living systems, of course, but it can also be made in a lab by chemistry. So we have these machines called DNA synthesizers, where you put some raw material in, something like sugars and some other things, and think of it like a typewriter with only 4 keys, A, T, G and C. So you type out a sequence, A, T, T, G, C, C, G, C, something, and then it's going to spit out a test tube with billions of molecules of DNA of that sequence in it, because it's a sequence, it's a string. The other nice thing about DNA is that it can stick with other pieces of DNA, not just arbitrarily, but in a very specific way. So A will stick to T, G will stick to C, and when they stick, you see this sort of shape. You see a double helix. So they will wind around each other and they'll form this sort of a shape. So going back to crystals, DNA is almost like a one-dimensional crystal. It has information in it. Here is one of the simpler objects that was made in that paper, and on the right you see the atomic force microscope image of that object. So that is a square. On the left you see a schematic, a cartoon of how that was made, and I'm going to explain that schematic to you. You see this one long strand that goes up, comes down, goes up, comes down, and goes all the way to the left. That is a strand of DNA. All these are strands of DNA. They've all been drawn with arrows, and these small horizontal ladders that you see, they represent the matching of sequences. So whenever I have an A on one, I have a T on the other, so they can stick. So how do I do this? I pick a long strand with some sequence I have no control over. Now I have to design sequences for all these short strands of DNA, all these short strings of DNA. What sequence will I design? I will design a sequence so that this short strand sticks to the long strand here and here. and for every short strand there is a different sequence. So in the actual structure there are about 250 different short strands. Here I have drawn much fewer, and then you go to your DNA synthesizer, you synthesize 250 different test tubes of DNA, you take a drop out of each, put them all into one test tube, you heat up that test tube and you cool it down, and in the process these molecules are all in a test tube, they are bumping around, and like these crystals, like these molecules formed crystals, similarly these molecules of DNA are going to come and stick to the long strand wherever they find a matching of sequence, and it's going to cause this long strand to collapse into the square, it's going to fold up into a square. So it's the same sort of thing, local rules, that I'm given a chart for the local rules, and in the end they enforce some sort of nice global behavior. So what will this form? Anyone? So I'll tell you what this is. You see this 1 denoted in a red arrow. So this is actually one strand, but I've drawn it on both sides. So it starts here, it goes left, it continues here, and it comes down. So it's one strand, but because I couldn't draw it on paper, I've drawn it as 2 pieces. And similarly for 2, 3, 4. So anyone, what will this form? Very good. So this forms a cylinder, and indeed if you do the experiment, you do see something like a cylinder. So what has happened here is the cylinder has rolled onto its side, and because of the way we image these things, there are some electrostatic forces that pull at the lower surface and causes material to be taken away from the top, and so you get this sort of cusp-like shape. So now we are ready to tackle a little bit of biology. So the philosophy is, you can ask where is the tree inside a seed, and we have seen that DNA is this really cool thing, it can contain some sort of program, and it can also act on this program and actually build things. In biology it seems that DNA performs a somewhat more modest role. It only acts as a store of information. It's a program, and that program is going to be executed by several other things. So every seed is made up of cells. That is sort of a very fundamental paradigm in biology, that all life is made up of cells, and inside every cell you have DNA, and this DNA is like a program that's driving the cell. And to execute this program, you have, like within human cells, you have about 25,000 different types of proteins. You should think, when I say protein, you should think of it as some sort of a nanorobot that can somehow interpret these instructions and run around and do very complex activities inside a cell. So a cell is a really complicated entity. Point of view is that the DNA sequence inside a seed is software for making a tree. It has all that information, and that information is going to be interpreted by other cellular machinery, and it's going to result in whatever it results in. And this is much more sophisticated than the examples we saw, like crystals, and even the last example with DNA self-assembly, DNA origami. But one believes that in principle it's still the same sort of process going on, maybe at a much more complex level. We started off saying that crystals could be possibly an origin for life, and that they seem to show some rudimentary forms of evolution. But is it conceivable that starting from something as simple as that, a few molecules, you could ever end up with the sort of sophistication that we see in the world around us? So one answer would be evolution. So whether you believe it or not depends on what precisely you understand by evolution. So consider this thought experiment. Suppose I have a DVD of Windows XP, and what I do is I take a million such DVDs, I randomly scratch them. What is the chance that one of the DVDs will have a better version of Windows XP on it? It's not going to happen, right? I mean, you just took some code that people have written with a lot of effort and you just randomly garbled it up. Even if you have a million DVDs, you're not going to get a better copy. And this is not, this is not irrelevant, because it turns out the human genome is approximately the size of Windows XP. So if you think of evolution as a purely random process that's just going to take your genome and just scrabble up the code in some way, then it's unlikely. You're not going to reach the sophistication necessary to build such systems, and once you have reached there, you're not going to stay there either. You're going to fall off. So let's take a small detour before we address this question. I want to ask a parallel question. Do human beings evolve at all? Maybe we don't know how they got there, but maybe once they got there, That's fine, they can stay there, they don't need to evolve. Okay, so think about this. Bacteria reproduce, they can reproduce very fast. They have very small programs, very small genomes. So it takes them 20 minutes to reproduce per generation. Human being, even a conservative estimate would put it at say 20 years per generation. And what does that mean? That means that bacteria have 500,000 generations by the time a human being has one generation. They can evolve much faster than the human genome. And so if the human genome were not doing something very clever, if it were not changing somehow, then its fate is at the mercy of bacterial evolution. So this sort of phenomenon that we saw, this phenomenon is, there is a competition going on here between bacteria and between human beings. And this idea is called the red queen hypothesis. It comes from one of Lewis Carroll's books, Through the Looking Glass. And so I quote, it takes all the running you can do to keep in the same place, somewhat like Mumbai, right, to keep running to stay in the same place. So what is it that I can do that's more clever? So let me sort of do a game here. Suppose, I have a basket of balls. There is someone who's trying to catch those balls. That person is very fast. And no matter where I throw the ball, that person runs and catches it. So what do I have to do to make sure the person doesn't catch all the balls? What if I take two balls and throw them at the same time at different places? Okay? Chances are he'll go for the one that is nearer, miss the other one, so at least one ball fell down. Right? So I want to do something similar. I want to produce variation in my population. Now there is a problem with producing variation. Think back to the Windows XP DVD example. How do I get another version of the same program to do something similar? Right? That's also not easy. As soon as I try to change something in the program, it might stop working. So somehow I want something that prevents a program from failing too soon. And so the idea is that I throw in redundancy. I throw in two copies of every program. Yeah, because there are two copies, if one of them goes wrong, the other one still works. So somehow I managed to get more variation in my population because of this trick. And each individual program can be in some sense less perfect, causing more variation. There is one more trick we can play. So it might be that when I have this DVD here that's sort of evolving, and so I have two DVDs together, right? It's a pair, and I have two DVDs together here, and they are both through changes I have done something to them. Now it might be that this pair of DVDs has evolved some very nice programs, and this has evolved some very nice other programs. But unless I sort of mix the two, it's hard for me to come up. It's good if I mix them together, then I can get one pair of DVDs that has the good qualities of both. I could also get the bad qualities of both, but that might be a risk worth taking statistically. So let us try to play this trick. So if this were DNA, it would be not zeros and ones but ATGC, but the same principle holds. If I have a program, in the end the program is represented as zeros and ones. So I have these two strings, two programs. I just pick a random position, I cut somewhere and I glue it. So what's going to happen? Well, this is what is going to happen. It's not going to compile. Right? You can't just take two programs, cut them somewhere, stick them together and hope that things will work as you want them to. So what is the solution? You have to do something more clever than that. So first I break my program up into small pieces, into small procedures, small functions, small subroutines, whatever you want to call them. And between these I want to throw in a lot of special strings that I recognize as comment strings. And then given two such programs, I'm going to search for these special strings, I'm going to cut at those positions and I'm going to stick at those positions. And all this seems very clever, very sophisticated, right? It turns out biology actually does something like this, and it's called homologous recombination. Okay, so let's talk quickly about aging now. So in any computer system, when you see any sort of behavior, you can ask, is it a bug or a feature? So has the designer thrown it in on purpose, or is it the case that it is just come about because of some other things the designer has done? And biological system is not designed as such, it comes about through evolution, but we can, with this analogy with computer systems, we can still ask this question, is there any advantage, look at the mechanisms by which aging is implemented. Is it an unavoidable problem or is it there for a reason? So there is some evidence that there are some cells and organisms that do not age and are potentially immortal. So this suggests maybe it is not an unavoidable problem. And if you look at the molecular mechanisms by which aging is implemented in cells, it starts to look very much like a feature and not like a bug. It seems as if it's been programmed in and it's not something that has happened accidentally. So then one starts to wonder, is there some benefit to aging? Is there a reason why aging has been designed in? And maybe I'll explore this later. Okay, so this is my last slide, intelligence. So fine, cells, we agree, let us say we agree that they might be similar to, they might just be some molecules coming together. But what about something as, as remarkable as the human brain? Could we say that the human brain is somehow beyond biology? Sorry, not beyond biology, but beyond the laws of physics, the laws of matter. Could it be something special, something extra? So is it that, human brain is intelligent, but machines can never be intelligent? So first, of course, we have to agree on a definition of intelligence, and this is a hard thing to do. One provisional definition of intelligence has been provided by Alan Turing. This is called the Turing test. Yeah, so that's Alan Turing's definition. It has the advantage that it's clean, it gives you something to think about. So here is one more quote that I like. This is by John von Neumann, who was one of the pioneers of the computer industry. He says, if you will tell me precisely what it is that a machine cannot do, then I can always make a machine which will do just that. So before we discuss that, let us think about this. What is a task? If you are really trying to distinguish a computer from a human being, what is a task that you would use? I can tell a joke, right? It's an easy thing. Human being might get that it is a joke, a computer might find it hard. Or music, I don't know, it might be hard for a computer to say that this is music, but that might also be a little subjective. So here is one thing that I thought of. If you give an image, with any sort of image, say a birthday party or something, and you tell the computer, now write a short essay on this image, describe what is going on. It's hard, right? A computer will need so much knowledge about the world to do something like this, whereas a human being would just do it, very easily. So is von Neumann really justified? If I give I tell von Neumann, this is my task, I'll give you a picture and I want you to write a story about this. So can he build me a machine that does that? Sounds difficult. But then the thing is that he uses this word, precisely what it is that a machine cannot do. So I have to tell him that precisely, which means whatever I tell him, he will say that's not precise enough. If I give this photograph, what will it do? Then I have to actually tell him the answer. So he will store it in some big table, and then he will ask, if I give this photograph, what will it say? Then I have to give him the answer for that. He will keep on doing this till he has a huge table where all the answers are stored, and that is his machine. So it's cheating in a way, but the principle, it's like Archimedes saying that if you give me a liver long enough and a place to rest it on, then I can move the earth. So it's a point in principle. He's saying that in principle I do not find anything different. In practice it's a very different matter. There are many, subtle things going on here. So when we first start to think about the difference between, life and matter, it seems that there is a very clear dividing line between them. On one side, we have these remarkable behaviors. On the other side, we have rather simpler behaviors. But if we start to think more carefully, it suggests that perhaps this line is not so clear, and perhaps it's blurring a little, or perhaps it is not even there. And life itself is made up of molecules. And it is these molecules behaving in all these remarkable ways that gives rise to the phenomena of life. But one still feels uncomfortable saying that life and matter are the same thing. So really we want to say that there is something more to life than matter. And for me the answer is that it is the sophisticated algorithms in living systems that are the difference between life and matter. Thank you.